and we're going to go through some of the conventional colocalization approaches, talk a bit about super resolution colocalization, and then finish on registration. So let's go through some of the conventional approaches. So in the old days, microscopes were diffraction limited, as in they could not go really below 250 nanometers in their ability to resolve structures. And as we'll see, this was good for colocalization, because we're able to resolve things down to basically chunks of organelle, and we're not able to go really down to localizing individual single proteins and things like that. So this is, when people think of colocalization, this is generally what the scale on which they think about. And this is how we're going to start, and then later on we'll talk about super resolution when we go beyond the diffraction limit and really start to resolve single molecules and small parts of structures. So I'm going to go through several colocalization paradigms, and I'm going to explain to you the rationale and the approaches that we'd use in each of these cases. So here we've got a cell, and it has in it fluorescence from two different sources, and we want to know about the relationship between these two different proteins or these two different structures. And this is what we call co-occurrence. Do the pixels of these different species co-occur? Are they in the same location? And the typical approach to this is the Mander's test, and I'll go through exactly how that works. But because we're sort of very interested in this overlap rather than more sophisticated analysis of the distributions, these are generally segmented, and we'll go into that. So it's a very kind of binary operation. You know, once thresholded, do these pixels overlap or not, and how many of them do? Okay, and that's in contrast to our second approach. So in this experiment B, we're interested in really the correlation between our two channels. So here we have rich intensity distributions in each channel, and we want to know, do these distributions have a similar relationship? So this is more than just overlapping. It's asking, are they high in the same places? Are they low in the same places? And so on. So this is a kind of intensity distribution analysis. Both of them work on individual pixel levels, as in we're not doing some kind of parameterization of the objects. We're comparing the pixels, but this doesn't require thresholding. Here we're looking at the raw pixel intensities in both channels and comparing how the distributions are. And this is really exemplified by the Pearson's test, which is a measure of correlation, and it's used for doing this colocalization. Okay, we will go into that in more detail. And then the sort of third-based approach is very much the object-based colocalization. So in this case, what we're doing is we are first parameterizing the cells, in this case organelles or proteins within the cell. We are segmenting and isolating each one and getting a coordinate location, a centroid for each structure. And then what we're doing is we're comparing these two distribution of coordinates. So there are other ways of approaching it, but what's similar to all of these objects-based approaches is that we individually parameterize the objects that we're interested in, and then we look at the relationships between these parameterized forms. With the first approach, we're still dealing with pixels, but they are thresholded. In the second approach, we just look at the raw pixels. And in this approach, we try and find structures in it and then compare those structures in a way which is we describe as being object-based. Okay, so I'm going to go over all of these in detail and show you how you can do them. So first, we'll start with the Mander's test. So imagine we've got this image, and you'll see this in the practical. We are interested in whether the green fluorescence and the red fluorescence are overlapping. And we're interested if this protein is located in lysosomes in this cell. Okay, so the first thing we do is we threshold each of the channels using something like UTSU, which you've already seen. So this is for channel 1 and channel 2. We could use a different approach for each channel as long as we're consistent for our experiment. So we always do the same thing to channel 1 and we always do the same thing to channel 2. And then what we do is we combine these using an AND operator. Now you'll see this during the practical. But what an AND operator is saying is, for every pixel, is it white in both of them? And if it's white in both of them, it will be white in the output. Okay. if it's white in one and not the other, then it's black, and vice versa. If it's black in the first and white in the other, it's still black in the output. But if it's white in both, then the pixel is both foreground in each, it's common in both, and you get a pixel here. And so if we then take this and overlap them, you can see that we have our channel 1 in magenta, our channel 2 in cyan, and then the AND bit, where they're both positive, is displayed as white. Okay, so these are the three things that we need to calculate the Mander's test. And so what we do, once we've created this channel B, which for me is both, we sum the pixels in each channel. So this comes to 519180 and 538560 in this case. And then when we do sum this, we get 30725, and it's no surprise to me that this is lower than these two, because these are the ones that are common to both, and so there'll be fewer than in each of these individual channels. And then the Mander's test is simply a calculation using these summations. So there are two coefficients for the Mander's test, M1 and M2. And M1 is channel B, so this output of our AND over channel 1, and M2 is channel B over channel 2. So what this is saying is, what's the ratio of the shared pixels over both of the individual channels? Okay, and through these two metrics, M1 and M2, you can then use that to describe your colocalization. So this is a typical kind of experiment. So I'm only showing you on one cell, but obviously you would process a population of cells from a number of independent experiments performed on different days and imaged on different days, and then you would amass the M1 and M2s from those cells and produce a distribution like this. And so what this shows you is that in condition A, so these could be two treatments from 50 cells, say, this shows that M1 has a much lower component of fluorescence. So as a ratio of the channel 1 fluorescence, the combined fluorescence is very low, whereas for channel 2, much more of it is shared pixels. So probably channel 2 has far fewer pixels in total, so the shared aspect of that is a much larger proportion. And then when we get to condition B, then we're seeing that almost a third of channel 1 is also overlapping with channel 2, and almost bit more, but a third of channel 2 is also overlapping with channel 1. So this is kind of a bit of a visualization to sort of show what that approximately would look like. And then we get a statement like this, which is, in condition B, the M2 ratio was significantly lower in condition A, suggesting that protein B had translocated out the organelle in which protein A was enriched. Do you agree? Perhaps, since we've already melted your brains, this might be a bit of an unfair question. But actually what's happened here is the red fluorescence here has actually shrunk, and that's what, in this toy example, is the reason for these coefficients changing. But don't overthink it too much. The point is, when you use the Mander's test, you get two coefficients, so you have to think a bit carefully about what that actually means in terms of your images. So you have to sit down with examples and make sure that you're interpreting it in the right way. But it's quite a powerful approach, and once you get your head around about what the M1 and M2 mean, and there's just these two simple ratios, then you can use it to do some quite powerful analysis. Okay, so that's an example of the Mander's test. And then I'm going to take you through a little bit of maths so that we can end up at the Pearson's test. I assure you it's all in your best interests. So for me, there are four basic operators in mathematics: plus, minus, multiply, and division. I personally feel like the dot product is like a fifth operator, where what you do is, when you have two lists of numbers, what you do is you arrange them together, and you multiply the first element of both, and then sum them to the multiplied second element, third, fourth, fifth, sixth element, like that. So we are multiplying each element in each position, and then we sum them all up, and that gives us our dot product. Okay, so from these two lists, which could be, we'll get to this, but this could be our channel 1 and our channel 2. To calculate the dot product, we multiply at each pixel, say, and then sum them up, and then we get one big number, which is our dot product, or sometimes known as the scalar product. Okay, here we're just demonstrating it on two lists of dimension 12, but if you did it on an image, it would be 512 times 512. It would be your whole image as one long list, and then one long list for each channel. Okay, but we'll come back to an applied example. Just be aware that the dot product is just this equation, and this looks more scary than it is. This is just the sum operator, and we're saying sum from I1 to n being the number of, say, pixels or the length of our lists, and then we multiply A1 and B1 and sum them up, and then this is it, just as an equation laid out in one line. Okay. And why, I've given you a bit of an intro into it, but why do we want to know about the dot product? Well, it's at the heart of a lot of techniques. And so if you really understand the dot product, then it opens up your understanding for how all of these techniques are working. So it appears in colocalization, which I'll show you soon. It occurs in fluorescence correlation spectroscopy, which I'll talk about on Thursday. Registration and colocalization are actually very similar, so it appears there. And convolution, when we're filtering, you're basically just calculating the dot product of your kernel and that position of the image. So if you understand the intuition for how this works, for what the dot product means, and I'll go into that, then it's a way into all of these areas and truly understanding them. So I mentioned that the two parts of our dot product could be an image, and just to reinforce that, what we've got here is a 2D image. And what we're doing here is we're just flattening it or reshaping it into a one-dimensional array, okay? So rather than having a 2D array, we're just flattening it into a long one-dimensional vector or array. Does that make sense? Yeah? So when we've got it in that form, we can start thinking about it as a distribution, okay? Now I want to break from that a little bit and talk about visualization of vectors. So here we've got vectors A and B. They only have two entries each, okay? A is 30 and A is 30, 30 and B is 40, 20. Now if we were to visualize that, it would look something like this, okay? So for A, we've got 30 in our first dimension and 30 in our second dimension. And in vector B, we've got 40 in our first dimension and 20 in our second dimension, okay? Now imagine we've got a vector the size of our image. We can't display it on a two-dimensional graph anymore. It would be a graph of more dimensions than you can visualize. So this is my fancy way of showing that. But basically, you can imagine 3 dimensions. So you'd have a vector pointing in an arbitrary direction in 3 dimensions. Now try and imagine thousands of dimensions which represent a different dimension for each pixel of your image. You can't actually visualize that, but hopefully you can follow the maths and where that's going. You've got some vector pointing into a hyperdimensional plane somewhere. Okay. And what the dot product means, and we go back to the 2D case, when you calculate the dot product of two vectors like this, it's talking about forget that for a sec. It's talking about the shared directionality of these vectors. So it's the proportion of them which is pointing in the same direction. So if we calculate the dot product of these two, we get 1800, and that represents how much of these twos sharing x0 and x1, how much of them is pointing in the same kind of direction. And to contrast that, what we've got here is two vectors which are perpendicular to each other, so they have no shared direction in terms of these axes, okay? And when you do this, you get a dot product of 0. So if two vectors are pointing in a similar direction, then you can calculate how similar that direction is from the dot product. Okay, and that's kind of the intuition for what the dot product is doing. It's telling you about the magnitude of the vectors in a common direction, the component of them which is in a shared dimensional space. Okay. So if we think about images, and imagine we had our vectors from these images, and we do the dot product on them, it's really telling us about how similar these images are in terms of those pixel distributions, like how high those pixel intensities are in the same locations. That's kind of what the dot product is telling us. The only problem with this is that the process is not normalized. So if you happen to have very high values in one vector, then it's hard to compare, because if one of your images is very faint compared to the other one, then it will give you a different dot product to the same image if it was just brighter but had the same distribution. So we want some way of normalizing this for the different intensities. Okay, so if you're still with me, so this is the dot product once again, which is great. But what we really want to do is have a normalized approach to comparing them. So rather than actually just using the dot product, what we do is we measure the angle between these vectors. So imagine we just had images of 2 pixels like here, then we can see if the angle between these 2 vectors, if we put them, if it's close, then it means they're similar. If it's 0, it means they're identical. Okay, so if the vectors have the same numbers in the different dimensions, then you get 0. The angle between them is 0. So this is a normalized measure of comparison. So why did I bring up the dot product? Well, it turns out that you can use 2 definitions of the dot product, the geometric and the algebraic dot product, and you rearrange them, and you can then extract the angle through doing that. So through calculating the dot product in the way that I showed you, rearranging it, you can basically get the angle out. So through the dot product calculation, you can get straight to this normalized form and compare the distributions using this equation. And that is exactly what the Pearson's equation is. It's the combination of the algebraic and geometric dot products. And when you rearrange it, you get this. R is actually corresponding to the cos theta, and it ranges between -1 and 1. And this is the measure of correlation, of colocalization. And I'll go into what the -1 and 1 mean, but it represents this angle between these 2 vectors. Now that's easy to visualize if you have very low dimensional vectors, 2-dimensional ones, you just plot it on a graph. But the same thing applies to these very high dimensional vectors, which would represent your image as a 1-dimensional, well, it's an array, but you take every pixel to mean another dimension. Okay? So if you can follow that through, and I know it's complicated, but it will give you intuition behind how convolution works and all kinds of things. But in terms of images, let's have a look. So here we've got 2 comparisons, both using the same channel A, but then 2 different channel Bs. We've got a CFP gamma 7 protein in a neuron. It's not so important. And then we've got early endosome A1 marker in the green here. And then we've just got dextran dye in the cell as well. And you can see when we compare these 2, visually they look very similar. And the Pearson's equation gives an output of 0.8, which is close to 1. And here, where they're not very similar, we get a correlation value, a rho, as it's also known, which is much closer to 0.2. And so I'll go through what that means. So if you have a rho, an R value of 1, it means that they are perfectly correlated. If you've got a 1, it would basically mean you've made a mistake, because it means you put the same image in twice, okay, for both channels. 0 means there's no correlation between the 2 channels, so they're random. And -1 means that they're anti-correlated. So this means that in one channel, the other channel is explicitly not there. Okay, so it's almost like, in this case, it would be as if the dextran had been forcibly removed from these organelles containing the gamma 7. Okay, and so some processes can do that. Certain proteins are excluded from certain organelles, and that's where you get a -1. But usually you get something close to 0, which means there's no relationship. And there's another analogy for this. So if you walk in a room and someone walks in at the same time as you, and then when you leave, they leave at exactly the same time, you're probably correlated with that person. So this could be your partner or something like that, and you have a correlation close to 1. If you walk in and out and they stay there, and vice versa, there's no relationship between the two, then that's probably 0. You don't know who that is. That's like someone on the tube or something like that. And if you have a -1, if you walk in a room and they walk out, and as soon as you walk out, they walk in, then you need to give that person some space, right? Because you are stalking them and they don't like it. This is anti-correlation. So that's one way of explaining the equation. There you go. So here's very much an applied biological example. So here we've got some cells, populations of cells, and they've stained, I think, some kind of viral protein in the red and LAMP2 for lysosomes in the green. And what they've done is a time course experiment where they've assayed cells certain minutes after infection by this virus. And you can see that the correlation starts kind of low at 15 minutes and then builds up to 90 minutes. But if we read the value at 90 minutes, we can see it's not massively high. It's like 0.3. So given what I've shown you, that wouldn't really, on its own, tell you very much about... if I just had 0.3, and forget this for a minute, if I just had 0.3, you'd be like, well, that's not very impressive colocalization, because I know that 1 is perfect and 0 is random. But as a biological experiment, you can see that over time the correlation has certainly increased, and it's increased statistically on this population of cells. So this to me is convincing, because there's been a statistical increase, and you can see what the colocalization was to start with. And this really exemplifies what colocalization is like in biological samples, in that biological samples are very noisy and messy, and there's lots of stuff going on. And very typically, the activated proportion of your protein that you're interested in is a small fraction as well. So most of that protein might be in a different organelle doing a different thing. There might be only sort of 10% or so which is phosphorylated and is actually doing the bit that you find which is interesting. So don't treat correlation value too seriously. You're always looking for comparisons against controls. In this case, it's a very nice control. They've got a different time point, and you can see how it changes over time. But you could also have a different treatment. Yep. We can see when we apply the colocalization to this image with this image, we get a Pearson's R value of 0.84. And this is fine, but in this image, our hypothesis is, is image channel A and image channel B correlated? Okay, and that's fine, but we get quite a high value there. And that's because the black pixels are also included in that calculation. And so if a pixel is black here, that's the same as here, right? It doesn't count as a 0 reading, which you get from, say, the Mander's test, because you're only looking at thresholded pixels, segmented pixels with the Mander's test. In this case, we're looking at all of the pixels unthresholded, so the black pixels will also contribute. As long as your hypothesis is that, as in, are the pixels in this image correlated, then it still makes sense. But we, as biologists or as scientists, are more often interested in what's happening in the cell. So in this case, we've changed our hypothesis. We've drawn a region around our cell. And here we are asking the question, are the pixels within the cells correlated? And now we get a Pearson's value of 0.66, which is lower, but it's more indicative of what's actually happening within the cell. So doing your experiment on the whole image could work, because as long as they're the same number of black pixels in each image, approximately, then you're still going to see strong relationships between your different conditions. But, go on. I mean, here we've drawn a region, but you don't have to draw it yourself. You could use one of these channels. The only problem is it's not always a great idea to use one of the channels that you're actually going to measure in a different way, because then you're losing that independence. Now, in biological imaging, people do it a lot, and you can get away with it as long as you're not particularly biasing your experiment by doing it. But if one of your conditions, the fluorescence was much weaker than in another one, then you could really affect your experiment by doing that. So you have to be careful. And so it varies from person to person, but sometimes we just get them to draw it by eye based on the bright field or something like that, because that's independent, and it also teaches them about the process and things like that. But ideally you would have a third stain, which we did in this case. We injected the cells with a dextran dye, and that allowed us to have a expression independent way of isolating the cells and then doing the colocalization. Yeah. What they did here I can't remember, but maybe they found the DAPI signal and did it that way. Maybe drawing a great, like a border around the DAPI signal or something like that. But just to get back to here, the real point is you still need to have proper biological controls to use something like colocalization. Although the Pearson's test has a statistical meaning, it doesn't really in the context of biology, because there's so much else going on in your typical image. So you always need to do the right controls and do comparisons between those controls with your conditions. Okay. But the Pearson's test is a powerful test because it doesn't just, you're not reliant on the thresholding. For one, it doesn't use thresholding. You may use thresholding to find the cell, but you don't use it for the actual colocalization itself. And it's also, because it looks at relationships rather than exact pixel values, it's very tolerant to things like global intensity changes. So one of the problems with doing stainings and things in microscopy is that from week to week, from experiment to experiment, your stain quality can vary a lot. And sometimes it's just brighter. Everything is just brighter, or it's just dimmer. The Pearson's test is very tolerant to global intensity changes. So what we did here is we've taken the same red channel in both images, but here I just added 50% intensity to this image, and you can see that the R value coming out is the same in both cases, because the change was a global change. And so what the Pearson's test does is it looks for, are we both high in the same place, are we both low in the same place, rather than did we both get to the value 253 in this channel, and vice versa. So it looks at relationships rather than specific values, and that's what makes it so powerful, and that's why it keeps getting used. But there is the caveat of the black pixels, which you have to be mindful and knowledgeable of to avoid that potentially influencing your experiment. Because you could see a positive correlation come just because one of your treatments caused the cells to contract, and that created more black pixels, which happens a lot. And then just to demonstrate anti-correlation, this is an image I've just inverted, but you can see how now the fluorescence is specifically excluded from these areas. And then when we compare that to the same, so this was actually the cyan channel here that I've inverted. I don't know why I coloured it red, and then I'm comparing it to the red channel, and we see now we get, rather than 0.66, we get minus 0.66. So it's just anti-correlated. Of course, you wouldn't do that. You would find a protein that was actively excluded from that organelle. But people rarely use this, so I don't have any good examples. But it could potentially be a very nice way of showing exclusion. Okay. So that's the end of the Pearson's test. Hopefully the dot product bit didn't blow your minds too much, but it's a very nice way of comparing raw intensity distributions. And the only caveat really is this black pixel thing, which you can avoid if you segment your cells. But it also means you shouldn't. I'll go towards the end. I'll show you a comparison of where each technique is strong, where each one is weak, and then I can point out some other good aspects of the different algorithms. But now we'll move on to object-based colocalization. So if you remember, this is where we explicitly parameterize each object that is there. So this could be mitochondria or some other organelle, or a foci or something like that. You just need to be able to isolate them from their neighbours. Okay. So here's an example of this. So we've got 2 channels here and a DAPI channel, and what we've done is we've thresholded the 2 channels, and then we've plotted lines between one channel and the other to the nearest object, and we're going to measure the distance between these and plot this as a distribution. Okay? So that was one comparison you could do. And here's another example where we've again thresholded 2 channels, we've found the centroid locations, and then we compare each location to its nearest neighbour and generate a distribution that way. Okay? But there's no one way of doing it, there's lots of ways. Once you open it up into this object-based approach, then you have to make decisions about how to approach it. Do you just take the nearest neighbour and then eliminate that from the other searches, or can multiple objects link to the same neighbour? What do you do if you've got more objects in one channel than the other? There are many ways of formulating how you do it, and there's no real one method as far as I'm aware which is correct or not correct. It opens it up to much more decision making on your part. This is work from my PhD, now more than 50 years ago. But what we did here is we identified these organelles that were moving down the processes of neurons, and we isolated each object, and then we just measured how many of them were overlapping. So we didn't measure the distance, we just said if you're within a cutoff distance, you are overlapping. What is the proportion of these fluorescent particles that are overlapping between all of these conditions? And that was a very simple but very strong way of doing it. Okay, so you can see there are a lot of black pixels here, and I didn't want to draw around the neurites. It was also, I didn't just want to threshold the fluorescence because I could isolate the individual structures, so that was a more powerful approach than just doing it on the pixel level. But yeah, we all identified the small circular structures, worked out if they're overlapping. I think it may be 50% pixels overlapping or not. And then we generated these statistics. And there were certain ones which had a certain level of just overlapping by chance. And then there were certain proteins that were clearly there was some kind of relationship between the two. So it convinced the reviewers at least. Okay. We're not actually going to do object-based in this week, but what you typically would do is do a distance transform between your different clusters, and then you could, in your next other channel at the centroid locations, you could measure that distance from the other distance transform calculated in the other channel, and that would tell you how far it was from each cluster to its nearest neighbour. So there are kind of elegant ways of doing it, but we won't go into it because there are a lot of degrees of freedom of how you approach that, and I'll talk about storm and palm analysis later, where that question comes up really a lot. So this is more to do with super resolution analysis, but there are certain people trying to make approaches which can handle all kinds of different situations, so it's worth checking out these papers if you're interested. But now I'll quickly summarize what I've told you. So I talked about Cooccurrence, the Mander's test. This works when one species is much sparser than the other. So you don't want to use the Pearson's test if you've got a lot of black pixels in one channel versus the other, right? Because then wherever you draw your cell, you're still going to have a lot of black pixels. So the Pearson's test isn't great if one of the distributions is much sparser than the other. But with the Mander's test, because you're thresholding, then you're just looking at the thresholded pixels and whether they overlap. And so this is much more tolerant to if one of the distributions is much sparser than the other. Okay, if that makes sense. Let's proceed. Again, so if there's a lot of black empty space in the analysis area and you've got no way of doing a region which just gives you your cell, then again the Mander's test is good. The negatives are it requires thresholding both of your channels, which can introduce problems because you might have noise and you might have a poor signal, and then you get artificially high colocalization. It's sensitive to noise, as all thresholding methods are. Then we go on to the correlation, the Pearson's test method. Very powerful, positives, very powerful analysis, no segmentation needed, highly resistant to global intensity changes, can be used for anti-correlation in a very easy way. but both channel species must occupy a similar pixel area because you're really comparing distributions, and if too much of it is zero for either one of the distributions, then there's no data there really for the comparison. You want to be positive for your distribution and also not saturated within all of the pixels that you're trying to measure, ideally. Yeah, and on that note, it requires the black empty space to be very low, but as long as the black space is similar in all of your conditions, then you can get away with it, but it's not great. And it's sensitive to noise as well, like random noise will throw it, so you might want to smooth your image or something like that before applying it. And then the object-based works when objects don't necessarily overlap. You can measure the distance between objects which are far apart from each other, and that can still be valid. And with the right object detector can be designed to be resilient to noise. So you'll see on Thursday we start fitting distributions, and that allows you to avoid a lot of problems with noise because you're looking for something specific and you have knowledge about what you're kind of looking for, but we'll cover that. And as negative points, it works very well when objects are small and uniform, but not if they're big and large, because how do you parameterize that in a sensible way? That's more for the other approaches. And it requires thresholding of both channels or some other kind of detection which can add parameters and things like that. So those are all the aspects. So really it's about picking the right tool for the right job at the right time. And that's just the problem. Yeah, you just have to learn all of them and then use them at the right time. But it comes from experience. Okay. At the start of the talk, we were talking about the good old days when microscopes were limited by the diffraction limit of light. But now we go far beyond that. We have STED, we have Palm Storm, and now we have min flux, which I think can get down to like 1 nanometer resolution. And this is amazing because now we can resolve almost down to, well, I think in case of min flux, down to the resolution of single proteins. But it requires a different approach in terms of colocalization. So you have to imagine that at one resolution, the good old resolution, then our intensity distribution was sort of continuous. It's on a pixel grid, but it's more or less continuous. There aren't lots of gaps in it. But imagine the same protein with a super resolution microscope, and you start to be able to resolve the individual clusters of proteins within that cell. And the problem with that is that from the simulation, you can see that when they overlap, in the good old days, then you get a high colocalization, but the same distribution but higher resolution, you get a Pearson's value of 0.23 because it's such high resolution that you can see when proteins are next to each other, but unless they're absolutely on top of each other, then they won't appear colocalized. So the use of our metrics kind of breaks down. It's very scale dependent, basically. And this is interesting and we have to do something about it. It's still possible to use some of the same metrics with, say, a STED microscope. And this is what we did in a paper, is that we found in peroxisomes, which is a certain organelle, that there were still interesting distributions within those actual peroxisomes. So what we did is we isolated the peroxisomes visually using a third dye, and then we just did the Pearson's test on each of the peroxisomes. So we got away from the fact there was a lot of black space, and in our case there was still enough overlap. The resolution was low enough that it was still meaningful to do our Pearson's test. And we did nice sort of controls where we flipped around the images and patches and things like that, and we could see that there was definitely a correlation there relative to the random controls and things like that. So although it's challenging to use the old metrics in the age of super resolution, it's still possible if you understand what's going on with the algorithm and how to apply it. But with Storm Palm, it's more tricky because with Storm Palm, and we'll talk about how this works on Thursday, but you are actually not isolating clusters of protein, distributions of protein like you do with STED. You're actually able to identify single emitters of fluorescence. And then what you typically do is you do some kind of clustering operation to link up the individual emitters, which are spatially nearby each other, either due to the accuracy of the system that they overlap in that sense, or they are close enough to each other to form what you think is a cluster in the cell. And once you've clustered your single molecule data, then you can start to think about colocalization in the conventional sense. But until that point, you're very much restricted to an object-based colocalization paradigm where every bright spot has been parameterized and you're basically measuring the distance between them. If you want to use the other approaches, then you have to do some kind of clustering and render it more like a conventional image. But there are many papers and approaches to doing this. It's not an exact science. So if you want to do colocalization and clustering, then I really encourage you to look into how other people have done it. But it's an expert thing that you need to apply yourself to. It's not. There are too many assumptions and decisions that you need to choose just to use an algorithm off the shelf. Okay. So apologies for that. So colocalization in the age of super resolution isn't as straightforward as it was. You have to really understand and learn them before going into that area, but that's what this practical is also about. But now I'll talk about registration. So before I showed you that slide where you have chromatic aberration and we want to know how we can correct it. Here's an extreme example where we've got a GPMV and you'll see this during the practical because it's nice, but we can see that the colors of this GPMV are not lined up. And what we want to do is realign these independent channels so that they overlap perfectly again. Okay. And I just want to talk a bit about registration. So what we do when we register two channels is that we want to find the transform which will make them overlap perfectly. So in this case, we'd want to rotate this and move it a little bit so that it would overlap in this channel. Okay. And there are many types of transformation that you can apply. So there's just translation, which is the movement of the channel, just moving those pixels X and Y. And then there's Euclidean with rotation. And then we start to combine rotation and translation and also perspective. Some of these translations are easier to measure than others. So in the case of a translation, we can just move our image around the pixel grid and see how it lines up. With the Euclidean, then we just rotate. With things like affine, then we have to do different distortions, which if you're searching a space of transformations, can be quite complicated to do. But for most microscopy applications, we can get away with, especially if it's chromatic aberration, we can get away with a simple translation transformation. And what this involves doing is moving one image with respect to the other and seeing how well they overlap in each of the positions, and then finding the position where they overlap the best. Okay. So I've got another keynote animation. So imagine these are our two channels, the red ring and the blue and the green, red ring and a green ring. And what we want to do is measure the overlap error as we move one relative to the other. So we're moving through each of these positions and we measure the overlap error, which is the gradient in the back in each of these positions. And then once we've gone through things exhaustively like that, we want to find the optimum overlap. So that would be this point here, and that's where the red and the green ring overlapped perfectly. So when you search through the space, you try and find that spot. And one of the main ways of assessing the overlap, a powerful method which appears again and again, is the correlation coefficient. And this is just the Pearson's equation, but in 2D. Okay. So what I was talking about, the Pearson's test, is absolutely applicable to this. Other than that, you just make sure that you've got X and Y involved as well, so you know exactly where you are. And that's why you should learn about the dot product and all that sort of stuff, because it keeps appearing. And this would mean that your images don't need to be of the same absolute intensity level, as long as they were basically high and low in the same sort of area, then you could get a good sense of how similar they are. Whereas if there's some reason one of your images was changing in overall intensity, you would struggle. So the correlation coefficient, basically very similar to the Pearson's test, is a great way of assessing how well your images are overlapping, and you'll see that in the practical as well. Okay, so in summary, some take-home messages. Colocalization is a popular technique which is used frequently in the imaging sciences. You have to learn to diagnose the needs of your experimenter to pick the right approach. Be aware that scale will influence your outcome and basically ruin your experiment. This is another important thing. Make sure people are taking images at the same resolution for the length of your experiment. Some systems allow you to change the magnification of your image far too easily, and if you've got images of different magnification or zoom factor in your data set, then it will ruin your experiment. So you have to make sure you're consistent for that situation, and understand your approaches so as to understand their weaknesses. Great, thank you very much.